Hi, everyone. Welcome to part one of our four-part digital event series called Confronting a Pandemic, jointly developed by STAT and Harvard Medical School. Thanks for being here. I'm Rebecca Robbins. I'm STAT's San Francisco correspondent, and in just a moment, I'll be joined by my co-moderator, as well as our featured speakers, to talk about new biopharma collaborative models for COVID-19. Specifically, we're going to drill deep on a partnership between Takeda and Novartis to try to develop a therapeutic known as a protease inhibitor to treat COVID-19. But before we begin, I want to go through a few quick housekeeping items. So at the bottom of your screen, you should see a few icons. There's a chat icon and a Q&A icon. So you can use the chat feature to chat amongst yourselves or to send a message directly to the STAT team. And if you find that you're having any technical difficulties, please let us know in the chat. And we ask that you please do not put your questions for our panelists in the chat. However, if you do have a question you would like to ask one of our speakers, we'd ask you to please use the Q&A feature to type your question over to us, and we'll do our best to answer it live. Again, we will not be taking questions from the chat, so please type your questions into the Q&A. So I'd now like to welcome my co-moderator, Stanley Shaw. Stanley is Associate Dean for Executive Education at Harvard Medical School. I'd also like to welcome our two featured speakers. Jay Bradner is President of the Novartis Institutes for Biomedical Research, and Andrew Plump, who is President of Research and Development at Takeda Pharmaceutical. So welcome everyone, and I'll pass it to you, Stanley, to kick things off.
Thank you, Rebecca. Thank you, Jay and Andy, and to all of our audience members. So as Rebecca mentioned, I lead executive educations at Harvard Medical School. And so we spend our time trying to catalyze innovation at the interface between academia and the healthcare industry, certainly biotech and pharma, but also digital health and many others. And so today's session, it's just a real pleasure to speak to Jay and Andy, two physician scientists who I've had the pleasure of knowing for many years and who are doing great work leading prominent R&D organizations. And we're certainly honored to be working with Rebecca Robbins and her colleagues at Stat News. I'm sure all of you would agree that Stat has really just done a wonderful job covering all aspects of healthcare, even before the pandemic, but especially during COVID. So welcome. Our goals today are really, I think, threefold. To talk about new collaborations and open science approaches that are bringing together companies in new ways to find new therapies for COVID-19. Second, to hear about efforts underway at Novartis and Takeda, whether it's repurposing or developing novel therapies. And third, to push Jay and Andy a little bit on what the new normal might look like for their R&D organizations and this new collaborative spirit. We'll start with a couple of questions on our side, but we'd like to take as many listener questions as we can. So maybe we'll start with you, Andy. Can you tell us a little bit about this collaboration and what was the impetus for it and how did it get started?
Great. Hi, Stan. Hi, Rebecca. Thank you very much for having me on the show. Jay, it's great to be here with you. Yeah. All right. So I'll dive just briefly into the collaboration that you're referring to between Takeda and Novartis, and I'll let Jay take that deeper. Jay called us one day and had this great idea. I went for 20 minutes talking about this idea. And at the end, I said, Jay, you had me at hello. But Jay will talk a little bit more about the collaboration that we're taking on together, because it's really one of many, and it's part of I think, a spirit that's evolving in the industry. I would say that when this whole thing came down in late February, early March, it was a really interesting time for all of us. And many of us were having individual conversations, pairwise conversations. All of us were getting deluged with emails from biotechs that had this idea or that idea, wanted to collaborate on this or that. And Jay was receiving the same emails that I was receiving, that all of the other R&D heads were receiving. It was just so redundant. So we started having conversations of how we could make this all more efficient. And in the second or third week of March, actually, Rupert Vesey called a handful of us together. You know, we're mates, we're competitors, but we're also mates. We've known each other for many years, and there's a great spirit of competition. We all want to win, but more than that, we want the industry to be successful. And so we got together on a phone call and we started comparing notes, and that rapidly turned into something that has been quite profound over the past couple of months. It's an effort called COVID R&D, and it's something that we can tell you more about. But one of the key elements of COVID R&D has been this project that Jay's been leading. And so when we get to Jay, I'm sure he'll dive deeper into it, Stan.
And so, Andy, before we get to Jay, so can you tell us a little bit about who's involved?
So, sure, and there's a Wall Street Journal op-ed piece today that talks about it, but it's secret, so it doesn't tell you about anybody that's involved, but it does go into some of the details. So it's not meant to be secret. It's not meant to be a secret society. It's actually meant to be a group of individuals who are together, not to talk or to drive bureaucracy, but to really get things done. Actually, the first phone call that I mentioned, it was Jay and me and Rupert, Matai Mamen at J&J, it was Hal Barron at GSK, and it was Manny Pangalos. And I'll never forget it. I was in my living room. It was a cold day in Boston, as most of them are. And we had the fire going, and we were all just kind of going off at all the different things we were doing. We were talking about shutting down at the time, which was something that many of our organizations were in the process of doing. And we just concluded simultaneously that we just need to be doing this together. We got actually McKinsey involved. They offered pro bono to help to coordinate because none of us could begin to think that we could coordinate anything like this. And we kicked off a number of working groups, one that we'll talk about extensively here, but several working groups around, how you run a master protocol today to conquer this disease, how you can set up assays preclinically to scroll through and screen compounds, and then some more forward-looking activities around data sharing and around novel therapies. And it was just extremely action-oriented, and it's been completely open. So over the past 8 weeks or so, we've expanded the participants. I don't want to say members because it's not really a society, but we've expanded the participants, and we've been really engaged, deeply engaged with efforts that have a bit more publicity, like the NIH effort called Active as one example. So we have no ego, no hubris. We just want to get things done in any way possible. So we do some stuff, and then we enable groups around us to do even more.
Terrific. So Jay, can you tell us a little bit about some of the first kind of early priorities for this group? And also, I know you have a history of open science efforts yourself, dating back when you were a fledgling kind of junior faculty member at the Farber, and you were mailing JQ1 out to hundreds of labs to let the ecosystem kind of share and collaborate. So how has that influenced you, and what is this coalition taking on?
Thanks, Stan. Thanks, Andy, and appreciate, Rebecca, the invitation to participate. There's quite a lot in your question. I'll be brief, but we can double click anywhere of interest to you or those logging in. Just exactly as Andy said, in January, February, we were getting reports back from our Shanghai site, and following the epidemiology of this burgeoning pandemic with a great degree of fear, but also a sense of accountability and a desire to respond. And as we looked across our organization at Novartis, admittedly first internally, we took stock in how we could contribute. We came up with these 4 concepts: drug access through our generics group, just to ensure that ICU doctors and nurses have access to the small format injectables needed to sustain life, the antibiotics to anticipate or treat ventilator-associated pneumonia. Immediate investigation. Could there be molecules that we have licensed in our broad commercial portfolio, or even investigational agents that we could deploy to study as potential abortive agents for the inflammatory cascade. Third, which I hope we get to talk about, is what Andy referenced, is discovery chemistry. The idea that there may not be a medicine on the shelf that can be repurposed with the type of efficacy required to really make inroads to treat this pandemic. The doomsday scenario would be that that the lack of sustained discovery science after SARS caught us off guard. And what would it take to stand up a bespoke and intentional oral pan-coronavirus inhibitor? And then fourth, contribution, just what can we do to support our communities? It could be PPE, RT-PCR machines, LCMS, a $20 million COVID response fund, as so many organizations in pharma and beyond have done. And leading research, I sat with my leadership team on this third thread. What did we have to offer that could be differentiating from all the wonderful responses we've seen, neutralizing antibodies, vaccines, and convalescent plasma, Andy, that your group is so expert in procuring. And we took stock in some of the platforms that we built over the last few years that were really robust. And there was this kind of convergence of having built this electrophile library to target cysteines and proteins together with Berkeley, a really expert and quite senior and accomplished team that studies proteases. And as you mentioned, this fascination with open science. And we started internally to generate immediately some momentum behind John Tallarico, who leads our chemical biology group in Cambridge, our global headquarters for research science. A team so senior we could normally never justify it, but everyone's in their studies like me right now. And we have the time. And I joined the team myself. I work in the assay development group. And it was amazing just to see how agile a big organization like ours could be when everybody had a real sense of a common purpose. And so the assays fell into place and crystals were obtained and, index molecules were studied. But we have some humility about this, Stan. Even, as big as NIBR is, we don't have a coronavirus expert on our team. We would need to look externally. As great as our chemical equity is, we had a small number of molecules biased for this 3CL, or so-called main protease, this cysteine protease that is an essential gene to the coronavirus. We had a small chemical collection for this family. And so through the group that Andy mentioned, this COVID R&D group, among the many, I would say, incredibly well-intentioned and effective ensembles of industry and academic leaders that have gathered in response to this pandemic. This COVID R&D group has proven unique because everybody that shows up has massive reporting lines and budget that they can deploy. And when we take a decision, individually or collectively or collaboratively, it just happens. And Andy, I was so thankful, I still am, just how quickly you stood up to say, we have protease inhibitors, we'll make them available, we'll figure it out, how to do it through our general counsels. And that really set the tenor. And other groups started to come forward with ways that they can help. If ever there was a time or a moment for open science, this is surely it. What the computer-assisted drug discovery field has done to procure fragments into the public domain, the massive use of pre-competitive publications and open publications, bioRxiv, ChemArchive, medRxiv, the flurry of response from journals to put knowledge into the public domain, often before it's fully considered. This is the moment, for open science. And build this program to, sort of leverage the scale that we can deploy for pipeline science, but make it accessible to anybody that wants to collaborate or honestly compete with us. What could be better than a race to make a coronavirus inhibitor when we're in such a moment of desperation?
So maybe we can explore a little bit more this main protease project. And could you tell us a little bit about how Novartis brought some existing collaborations, some pre-existing probes or expertise, and then I'll throw it to Andy for Takeda's efforts and what they brought to this protease project.
Yeah, no, thanks. Well, I mean, Takeda doesn't need Novartis to make a drug. Let's just be clear that we all have capabilities in our interdisciplinary scientific environments to operate singly and competitively, and we relish in that. But we do often collaborate, combination clinical trials. There are plenty of models for this. In our case, we have a group in Emeryville so expert in complex protein crystallography, two-dimensional NMR, fluorinated libraries for fragment-based drug discovery, protein biochemistry. It sits right next to Berkeley, where with Dan Nomura, we've started to build and study the ligandability of cysteines in the proteome, to map cysteines in the proteome, that we built out a team there that could leverage all of the incoming genetic information from clinical isolates to make sure that we had MERS and SARS and SARS-CoV-2 and even emergent polymorphic flavors of this protease purified with assays developed in Cambridge and Basel. By having this kind of core capability in lead optimization and understanding the mode of molecular recognition with crystallography, we could take an input of chemistry, probably from anywhere, and assess it quite rapidly without anyone having to build these capabilities. And so we've prosecuted a few thousand molecules from our library. We've started to input computationally predicted molecules from CAD, computer-assisted drug discovery. And we're just now at the phase where we have the pipeline so performing that we can now start to bring in these biased chemical libraries, these focused libraries from Takeda, and turn that data around to Andy, structure blind, for his scientists to look and decide what may be actionable. We intend to make all of these capabilities accessible. We're happy to deposit the protocols for these proteases into the public domain, into contract research organizations. Maybe people don't want to collaborate with Novartis. That's okay too. They should be able to access these learnings to expedite drug discovery.
Great. And so, Andy, how has this dovetailed with some of pre-existing interests at Takeda and your platforms and expertise?
Great. I mean, I think similarly to Jay and to Novartis, Takeda has a deep interest in partnerships and in trying to identify new ways of working. There's just so much inefficiency and redundancy in our industry at baseline. And while competition is a main driver of forward momentum, we just need to find a new way to work, because there's just too much background noise and there's too much redundancy. There's also situations like this where there's a crisis, and we've seen this in the past. There's a lot of energy and enthusiasm, resource commitment, and 6 months later, when the crisis dies down, everybody moves on. And so I think Jay, and in this project in particular, and us, saw the potential not just to leverage partnership to get things started, but to create momentum so that we could push this forward. Because let's face it, in 6 or 12 or 24 months, I mean, we're all imagining that we're going to be in a very different place with respect to this particular pandemic. But as Jay noted, this is something that we will have again and again. We've seen this now 3 times over the last 2 decades, where a member of this coronavirus family has leaped from animals to humans. This will happen again. It was actually, just commenting, one of the meetings that we were at, the COVID R&D meetings, Jay got up and started to show this protease, and he did an overlay of the SARS protease and the SARS-CoV-2 protease, the 3CL, and just layered them on top of one another. And they're 95 or 97% identical overall, but he showed the active site, 100% homology in the active site. And it was striking, and Jay, I think you even mentioned this, to imagine that had we continued efforts that had initiated 15 years ago to make a molecule that would have inhibited the protease for SARS, we would have had a drug that was tailor-made for CoV-2 today. So we have to make sure that that doesn't happen in the future. So we're completely on board with that. We didn't have an active effort in the space. We have a library, as with many of the large companies, our chemical libraries are massive. they're largely overlapping, but they're also largely unique. And that uniqueness just derives from our history, right? We all pretty much start off with the same starting material, but then the libraries evolve and diverge. And so we've had a library that, because we are, historically, we've been a small molecule chemistry company that has focused on proteases in the past. And so we had a protease-enriched library. So we were taking on other efforts at the time. Our head of research, Steve Hitchcock, who's an amazing guy, fully committed to this. You know, as soon as Jay called, I was excited. I called Steve and he said, Andy, you had me at hello. And so we jumped in. We have a close relationship with Schrodinger, which does in silico modeling. We also have a very strong structural biology group. And so we started to screen in silico compounds. And I don't know, Jay, we sent out something on the order of 10,000 compounds to put into this effort to be screened. So we're all in here. And you know, I think the commitment that we can make now, because we're public, is that it's not a 6-month project. This is a multi-year project that we're committing to.
So I want to jump in with our first audience question, which pertains to something Jay had mentioned earlier with quite a great deal of optimism, and that is sort of how preprints and the rush of information has sort of fueled an advanced open science. But we've got a question from Denise Silber, who poses kind of the other side of that issue, which is that the mushrooming of preprints that we've seen from COVID, and these are articles without peer review, can sometimes inspire lower trust. And so Denise asks, is this too the new normal?
You know, I hope that the use of preprint servers, which, you know, debuted, of course, years ago, and it's been the dominant way that physicists and mathematicians communicate for decades. I hope this is the new normal. I have many colleagues still in academia who've shared with me that they had maybe an experience of a handful of articles, a graduate student who've, you know, got religion about open science, or themselves wanted to test the waters to see if it's averse to the competitive publishing environment. But now it's emerging as the norm. And I hope that this remains. There indeed are challenges. When I read a bioRxiv paper, I read it with a reviewer's mindset, critical mindset of what I'm seeing and what is being messaged. One of the advantages amidst this SARS-CoV-2 pandemic of the preprints is that we're seeing a lot of different papers emerge immediately on the same subject. And ultimately, the best reproducibility is the independent validation experimentally of the same focused idea. It means you got to read a lot of papers. And it is quite a volume that's coming forward. But I should say that some of the studies that we've seen in structural biology, some fundamental contributions to assay conditions or substrate sequences, really did get us started to test the flavors of the protein that we were purifying in the laboratory. So there has been a tangible acceleration to this project, as well as all of science, even amidst the complexity and uncertainty of pre-peer-reviewed disclosure.
And we've got another question from Paulina Rychenikova. And Paulina asks, what is the best way to join one or more of the working groups that you've described with these two companies and other collaborators? At Argonne National Lab, that's a DOE lab in Illinois. The team there is very active in computational drug screening efforts and antibody design. And I think many of our listeners on this call may be interested in how their organization could potentially get involved.
Maybe I'll start, Jane, if you want to add. So one of the groups that was involved in COVID R&D right from the beginning was BioSentry, and actually BioSentry put up a link to it's a portal. It's an information sharing portal that connects outside groups to COVID R&D. I mean, you can certainly contact any of us. You can contact any of us directly. What we were trying to do through the BioSentry portal was, in a sense, expand the concept of open innovation to open information sharing. So you have access to everything that's going on within COVID R&D, but you also have access through that site to a much broader array of activity that cuts across multiple different alliances. I would really encourage you to try to connect with something that's active rather than starting a new activity. Because one of the things that happened over the last couple of months, all with good intent, was this mushrooming of alliances. And my name was showing up in 8 different places, and I was supposed to be on 8 different phone calls every other day to discuss exactly the same topic. So we've been trying to cut back a bit on some of these distractions. So rather than starting something new, my guess is that if you have something that's interesting, there's probably already an effort started and already an interest in partnership.
Yeah, I can only agree with what Andy's saying. When we had this JQ1 experience back in the lab at Dana-Farber, it was very interesting because GlaxoSmithKline also innovated bromodomain inhibition and had a network of collaborators, and having more than one node in a scientific space, even as narrow as a single gene product as a target, can have a multiplicative expansion of ideation and of experimentation. I would hate to think that, as excited as I am about what we've built around the main protease, that we would become or could become a bottleneck to this science, as opposed to putting these methodologies out there. Now, with that said, as you're from Argonne National Labs, and as I'm like a huge Cubs fan and a Chicagoan, just give me a call. We'd love to work with you.
So the collaboration that you've described is sort of taking place under very unusual circumstances, of folks working at home, trying to coordinate these things via Zoom call. And so I think many researchers, all of you included, are trying to adapt to kind of a different communication style. And to that end, we've got a question from Michael, and Michael asks, are there any new learnings or ahas about how to use data or digital tools to conduct studies in this environment?
Well, for sure. At this time, it's become very hard in drug development for us to safely visit sites, hospital networks that really are restricting external, even collaborators on clinical investigation. It's been hard to do patient visits. And we've pivoted just immediately to remote monitoring and new ways of executing in drug development. Andy, you've been leading an effort to try to pull data together from control arms to try to even compare one study to another as a benchmarking, or in aggregate, really understand what does it mean to be a hospitalized ill COVID-19 patient with or without respiratory complications in this geography versus another. The nimble response by clinician scientists has created a somewhat of a complex milieu of interventions, sometimes within an investigational framework and sometimes beyond. And so I actually think that connecting through data and leveraging some of these digital capabilities is a learning that we're all getting used to. We had probably experimented with it like most others, but now that it's become our uniform practice, and we're losing very few patients, thankfully, to follow up on our studies, and whereas new study starts are hard, we're continuing to enroll even difficult cancer studies. I think that these are the types of learnings that will stay with us long after this pandemic ends. Andy, what would you say?
Well, I agree fully. I think the technology accelerates rapidly to use in a research lab setting. In a clinical setting, that's not so much the case. The inertia that develops around existing ways of running clinical trials is very hard to break. The trials are really expensive. They take a long time. They already have immense risk. There's a huge reluctance to embrace new technologies. Huge. It's really tough to penetrate. On the flip side, technology is there. Technology is not the issue. It's really behavior. We have a huge opportunity to take advantage of the situation that we're in. Hopefully in 6 or 12 months, when we're really back up and running full steam, we're doing it differently. I've suggested internally that when people ask, will things slow down? The answer is for sure over the next year. If you look in aggregate at our industry, clinical trials will slow down. my guess is in 3 to 5 years, if you look at everything that we're doing now and you add together what we're doing in the future, leveraging some of these more efficient tools, in aggregate, we will be going faster.
So can I just follow up on the clinical trials? So with regard to COVID-related clinical trials, you know, there's some encouragement from WHO or FDA about, you know, master protocols. Are you speaking and working with multiple companies to try to get these master protocols going?
I would say yes and no. Andy, why don't you take yes and I'll take no, even though I'm sure we're both
that plays to our personalities, Jay.
we're both doing both.
Yeah.
So,
well, I get yes and you get no because you're a Cubs fan and I'm a Mets fan.
exactly. Oh, we're getting along so well. No, there are great efforts to stand up master protocols, and Andy, to your credit, is leading one of them. At Novartis, we started to worry in February and March with the prolific use of hydroxychloroquine, that there wasn't a data set around it really to assess efficacy by the standard that we're all used to. We manufacture hydroxychloroquine through our generics unit, Sandoz, and we donated 130 million doses just so that it would be accessible with this incredible demand that was arising. And in some cases, it was investigator-initiated trial need, but you know, some countries were putting this into treatment protocols. But we didn't feel right just making the medicine available. We wanted to study it as well. And so thinking about putting together a master protocol and bringing in multiple arms and having, honestly, through that perhaps some efficiency versus just buckling down, writing a phase 3 trial, taking it through our governance by circular and opening it globally, it proved a little bit faster. And this is not in any way to slight the efforts towards master protocol development. But there are some cases where you have a focused question and you just want to get it answered. And with a global clinical trials organization as we have, we found for that and 2 other phase 3s that it'd be much faster just to stand them up as concrete protocols.
And you know, I'm sorry, just on the master protocol thing, I think it also gets to ways of working. And I think, you know, Novartis is a great example of this is how we get things done, and they got it done. And so it's another reason why there's a reluctance to change, is because in many places, the old way, the established mechanisms work really well. And I think, though, that when you step back, and this is something we've had many discussions around, if we don't leave this crisis, if we don't work through this crisis predominantly through master protocols, and if we don't leave this crisis having as a tool for us to leverage in the future the master protocol concept, I think we will have failed as an industry. I mean, it just makes no sense. In short order, we'll have 12 different neutralizing antibodies that six or 8 or 12 different companies are making. If each of those are being studied one-off in each of their own placebo-controlled or standard of care controlled studies, and they're not being studied head-to-head, you know, shame on us. I think that we need to come together, especially at a time like this. I mean, there may or may not be a business opportunity here for individual companies. Maybe, maybe not. This is a perfect opportunity for us to come together, enhance efficiencies, and figure out new ways of working.
And just for any of our listeners who might not be familiar, could you explain exactly what a master protocol is?
Sure. So different constructs, but it's essentially saying, as opposed to taking a control arm and an active arm and then running a study, you take one control arm and multiple active arms. So you increase efficiency because you have a single control arm, and in many cases you allow comparisons between active groups. If there are more sophisticated ways of running these studies where you can do what's called adaptive design, where you bring in an active arm and you let it go to a certain point, and using disciplined statistical methodology, you make decisions as to whether you think it still has a chance, and then you allow it to continue, or you take it out, and then you allow new drugs to come into the protocol. So it's a really efficient, and if we can get our act together and operationalize these protocols, which is something that's not easy for us because we've really not done them in the past, if we can make that happen, it's a much more effective way of assessing medicines in a variety of different diseases. It's done, in the area where Jay comes out of in cancer, the field's been much more creative in using these types of designs for a variety of different reasons. We have to take that an additional step in cancer, but also in other areas.
Yeah, and to be clear, we participate in master protocol studies in cancer and beyond. I think in the signal finding space of proof of concept studies, this is a very powerful way to generate a very convincing data set as a bake-off of certain technologies. In the phase 3 space that we try to stand up these 3 protocols, there are some little design challenges. One of the medicines is going to be subcutaneous. Another one of the medicines is going to be oral. And so your placebo arm then has to both take a pill and get it injected. It starts to become a bigger study, believe it or not. But in signal finding, as Andy's saying, these are very powerful instruments, and there can be a big learning standing up these protocols amidst the pandemic.
So for our next audience question, I want to look backwards to kind of before the COVID-19 era. And Esteban Dominguez asks, quote, before the pandemic situation, were there any other examples of this sort of collaboration? What was the kind of difficulties that may need to be overcome so that collaborations like these can happen and continue and flourish? So I want to ask Andy and Jay if there are any kind of insights you've gained from trying to do things like this in the past.
Andy, I don't know about you. I really like working with the IMI. We've had a great experience with the Structural Genomics Consortium. I think that there's a legacy of very positive public-private partnerships sponsored by the NIH as well. And so from an industry standpoint, and I've experienced it as my prior academic life as well, there have been some really stellar examples. In the SGC case, structural genomics, is about creating chemical probes for new protein targets to just sort of jumpstart discovery chemistry. And many, many chemical probes emerged out of that effort. The same was true in crystallography, for that matter, around 3-dimensional coordinates. Through the Innovative Medicines Initiative in Europe, we're a very active member, participating in probably a dozen public-private partnerships, each focused on a specific fundamental challenge. Can we combine all of our tox data to start to build a computational predictive model for small molecule toxicology that would allow us to do animal experiments in a more sparing manner, or more efficiently imagine what the next molecule in a chemical series might be, and things of that ilk. So I do think that there is a groundswell of evidence that these work. On the flip side, they can be bureaucratic, they can be slow moving, they sometimes don't get the attention that's commanded by a pandemic. Science by committee sometimes is not as powerful as science by the committed.
So the pandemic is affecting the globe, but as we've seen so far in this era, sometimes individual countries and states are sort of operating separately and sometimes competitively. And so along those lines, this next question comes from Jeff Roy, who asks, broadly, how do you anticipate state security interests will influence your collaboration and progress? Do you see barriers to working across certain environments? And on the more optimistic side, has the crisis created new opportunities to enhance global health throughout your organizations?
Andy, you want to start?
I thought you would jump all over this one, Jay, but I certainly can. So interestingly, we're a Japanese company. We're a 239-year-old company that has been based in Japan for that entire 2.5-century tenure. Over the last 5 years, we've moved, and we've recognized that the center of biotech innovation is in the U.S. and for the most part in Boston right now. And so we've really rebuilt our research laboratory and our development group out of our Boston area. But we're still, and Novartis similarly, Jay can tell a similar story as a Swiss company, but we're still very committed to ensuring that medicines are accessible to patients wherever they are across the world. I think, but the nuance of the question is that we're living in a time that's quite different today than it was even a year or two ago, with nationalism and sentiments sweeping the world even before the pandemic, and now exaggerated by the pandemic, that creates a lot of uncertainty, actually. I'd love to think that as we think about this pandemic and the challenges that we're facing, that we're first thinking about patients with greatest need and not trying to satisfy the full needs of a country, but trying to satisfy the needs of patients across the world. If we end up with medicines, my hope is, in the end, there will be issues of supply, certainly at the early time. And I hope, and I bet that many would agree, that when we look at where those medicines should be distributed, we're not looking within the boundaries of a country, but we're looking at within the context of unmet medical need, and that the patients with the greatest need are receiving those medicines and those vaccines first.
Yeah, I agree. I think that this pandemic shows us something we already knew, is that the globalization of science is so radically distributed right now. I mean, just today I read a stellar biochemistry paper from Poland, a crystallography paper from China, all on the subject of COVID, all emerging very much in real time. So knowledge transfer and data transfer in biomedical research science right now is liquid fast. Faster perhaps, and perhaps even more reliable than some epidemiologic data. The challenge is the passage of scientists, not their science. We're all under very tight travel restrictions. We've had to reimagine how we gather, leveraging Zoom and other formats just like this. It's just not exactly the same as shoulder-to-shoulder whiteboard ideation. And so I do look forward to getting back to traveling and being with our chemists in Basel and being with, collaborators at biotechs in Northern Europe. So that has been, I suppose, a little bit of a barrier. I do hope that, with all the complexities of a Brexit and the way that post-COVID China and Beijing and Shanghai are hard to access physically for scientists, that we'll get back to a smoother traffic of scientists between geographies.
So this next question pertains to pharma's reputation, and it comes from Rose Hoban. And Rose asks, many of us are already starting to see and hear anti-science, anti-vaccination, anti-pharma rhetoric springing up, and some of it can be really eye-popping. And Rose notes that, pharma, of course, is often viewed skeptically in the public. And so in your efforts, have you identified a strategy to help the public feel comfortable with whatever ultimately comes out of this collaboration?
I have in the background the Pharma Reputation Index, and just in the 45 minutes that we've been on this panel, it's been ticking up. So that's a good sign. So, but in all seriousness, I think it's a big issue, actually, and our reputation is horrible. It rivals the tobacco and the oil industries, and it's sad. I think a lot of that we brought on ourselves over the years with behaviors starting at the turn of the century, marketing behaviors that shame on us. I think a lot of those have been turned around, and if you look inside the walls of large pharmaceutical companies, for the most part, you find people who are deeply committed to doing good and to improving the lives of patients across the world. There are, however, bad actors in the system that are driven by greed, by ambition, and by avarice, and unfortunately they pull us down. I think we have an amazing opportunity here, not by using PR, but by doing the right thing to really help to turn that reputation around. And the commitment that you see from partnerships like Jay's and mine, we don't see this as, we're not doing this for profit. We're not doing this to drive directly our underlying business. We're doing this because we need to do this. And there are a dozen other R&D heads across pharma and dozens, maybe hundreds of leaders across the biotech and healthcare industry that feel exactly the same way. It would be a shame, again, if we leave this crisis and haven't repositioned ourselves as leaders of healthcare in the world.
Andy, what you said, I used to work at a cancer charity before I came to NIBR, which is very much the core discovery engine of the Swiss pharmaceutical giant Novartis. And it was an adjustment to be the very same person, wired the very same way, caring desperately about patients and and trying very hard to find creative solutions, and then to land in an environment where no one's cheering for this science. I think the public is right to hold the pharmaceutical industry to account in exactly the way that Andy so eloquently said, but I think wrong to hold it in contempt. The scientists that I work alongside are wired just the same way as the academic and government scientists. I think that this can become a very important time for science. I don't know that this will change the reputation of the pharmaceutical industry. I do know that my mom, who is, an 80-year-old accountant, can spell remdesivir according to a recent email, and that's a huge advance. I think that people are celebrating nurses yesterday. I think that, I think this is a time when people will realize how vital having an authoritative scientific industry really is. And hopefully we don't end up distracted by academia versus industry and large pharma versus medium versus biotech. There's great advances happening everywhere. I have learned to maintain just sort of a quiet and personal sense of pride in this work that's often not celebrated in the way it was in academia, and that's okay at the individual level. But I do hope that this is a wake-up call for society, that the investment that they have made, that the public has made in almost every country to stand up a strong biomedical research environment can deliver in moments of total global desperation.
So for this next audience question, I want to pivot to the regulatory side of things. And Jeff Lazar asks, will the new normal result in the FDA being more willing to accept both large, simple trials as well as actual use trials? And if you think so, do you believe that the appropriate statistical tools are in place to usher in some of these new approaches?
Well, maybe I can start, Jay. So, well, certainly during this, the question I think is projecting into the future beyond COVID, but I think we need to look at the incredible and heroic efforts that the regulatory agencies, and in particular the FDA, are taking during this time period. They're working nonstop and accelerating everything that's COVID related. You saw with remdesivir an overnight approval for emergency use authorization, and that's been the norm. Actually, we have a program it's an early program. It's not been in very many patients, but it's an oncology program for which we think there could be rationale to test in oncology patients with COVID. And we sent documents in on a Friday. On a Saturday night, I was on the phone with the deputy director of overall of CDER, and then Sunday morning with the head of the oncology division, because they're just so engaged in wanting to push things forward. So, really hats off to everything that they're doing. And, despite some of the criticism that they've received, they really are focused on science, and they're really making data-driven decisions with thinking about patients that are affected first and foremost. I really believe that we have an opportunity to leverage this and to change our future. Now, whether real-world evidence generation can ultimately lead to changes in practice, that's something we still need to see. It's a really tough space. The benefits of the randomized clinical trial are just immense. There's huge opportunities, though, to change how we approach those trials. We're already doing that, in particular in oncology. Many oncology programs are registered on tens or small numbers of hundreds of patients. We already do that. Can we extend to other areas? And then beyond that, using real-world evidence data sets to bolster, let's say, our ability to use medicine across indications, I think absolutely.
So can I, we've alluded a little bit to the future. So if we project forward, how hard is it going to be to continue this entrepreneurial and collaborative spirit and this focus, to prosecute a COVID or coronavirus therapy, to its conclusion? I think both of you described this conversation between you that said, had we finished the job, with MERS or SARS, we might have a therapy. And so you were doing great, both of you, but you were under a lot of pressures. You have to manage your portfolios. How hard is it going to be?
I think, Stan, this is a really important question. I think it's probably the toughest question for Andy and I to answer. If in 2003, in the wake of SARS, the National Academies got together, they wrote a report. It's hard to read. It's hard to go back and read this report. It's publicly available. It spells out exactly what's necessary should SARS reemerge. It's the playbook for exactly what technologies, what vaccines, what direct therapeutics, the main protease inhibitors, chapter 4. And this work was initiated. There was a blush of high quality scholarship in the wake of SARS, but then it dissipated. Perhaps SARS with 8,000 infected and a 10% mortality rate didn't send the shiver down the spine of large organizations, academic or industrial, that the present pandemic has done. And now here we are with SARS-CoV-2, ill-prepared, but mobilizing unbelievable resources across the industry to respond. So there will be a blush of scholarship and there will be a blush of innovation. What Andy and I spend a lot of time talking about is what makes this sustainable? What's more sustainable, open science or competitive science? What's more sustainable, science for the public good or science that has a profit motive that can survive tough quarters or portfolio decisions? I hope that we're able to stimulate a competitive spirit in the industry writ large, that someone will make an RNA methyltransferase inhibitor, an RNA-dependent polymerase inhibitor. We'll take on the protease inhibitor. We'll end up competing with 3 other groups. Because in those spaces where science gets intensely competitive, AAV9 gene therapy, CAR T-cell therapy, immuno-oncology, it sustains itself. I'm not worried in this case that I will be able to sustain this thread of investigation at Novartis, because baked into our strategy is the Novartis Institutes for Tropical Diseases. And we have coming next week for approval this project as a formal pipeline project. It will be reviewed, but once approved, this project sits at the NITD alongside anti-malarial programs that have managed to weather the storm of 5 or 6 serial restructurings. But Novartis alone won't cure coronavirus. And so I think that you're right to ask this question, what makes this sustainable? And Andy, it's you and I will need to hold ourselves, our organizations, and our colleagues' organizations to account.
Andy, any comment or?
I mean, just fully agree. And I think, it's easy for Jay and I to say we're all in and mean it. And I know that Steve Hitchcock, who's our head of research, is committed to a full lead optimization program. We're going to make a molecule here and we're going to bring it forward. I think actually the cost for doing that, it's not massive. It's doable. We also have to imagine that there'll be new structures in place. After 9/11, we had the Office of Homeland Security that was built. I'm imagining, we'll see if this happens, but I'm imagining that from a public health standpoint and a national security standpoint, there'll be new structures in place that will enable some of what we're doing. But I think where you're getting to, Jay, which I think is the big question, isn't whether or not we complete the activities that we're starting on some of these individual targets, but whether we can sustain an effort that's forward-looking, that's trying to predict what that next pandemic is going to be, and that's creating an infrastructure, the right level of resourcing to support what's necessary to treat that. So we're in on the protease. We'll have an inhibitor for the protease. I don't care if it comes out of Novartis or Takeda or wherever. Just want to make sure that we get that. I know Jay feels the same way. I think it's thinking beyond that, and that's going to require a dialogue that we continue well beyond this pandemic.
And one last question from a member of our audience, and that is, might this model work beyond the world of infectious diseases? Might this collaborative model be adapted for oncology, for instance?
I'm smiling because at the end of the second meeting of COVID R&D, Andy, I don't know if you remember this, the first meeting, we're all introducing ourselves and what our roles are and what our organizational response is. And the second meeting, we're starting to get a flavor for work streams like master protocols. And it got so granular so fast. It got right down to the project level, you know, this is what we're doing. I've got this study we're standing up, and on and on. And at the end, when we're closing the meeting, I said, man, let's do this again on Alzheimer's disease. because we don't, you know, we don't immediately rush to reach out in the earliest moments of drug discovery. And by the way, there's good reasons for that around competition and antitrust and other reasons, you know. But I read an article the other day that a company that makes a cell therapy was going to collaborate with this other tiny little company that makes a non-chemotherapeutic conditioning regimen. And it was such a breath of fresh air to see companies in early space, you know, team up, which almost never happens. You know, when the chocolate finds the peanut butter, like amazing things happen. And so it's a great question. It's on us, Andy, to find, I think, an equally monumental challenge in the wake of the current and commanding challenge, to think differently about how we action science.
Well, with that, I think we're about at time and need to wrap things up. Thank you again to Jay and Andy, and to Stan and Harvard Medical School for working with STAT to produce these chats. Our next chat in this series will be taking place on Tuesday, May 12th, that's next Tuesday, at 1 p.m. Eastern Time. STAT and Harvard Medical School will come together again to talk with experts about telemedicine in the time of this pandemic, and I hope you can join us. Thanks again, everyone. Stay safe and be well.